Let me share my screen. Somebody's sending a chat message. Let me warn you that when I share my screen, I won't see any of the chat messages. So if you have a question, please ask it out loud. All right, you should be seeing my screen. Can somebody confirm that you're seeing my screen?
Yes.
Yes.
Okay, thank you. That message doesn't make sense, so I'm not going to leave it. Good. All right. So today we're finishing chapter 1, and we will continue with chapter 1 a little bit next Tuesday, and then start chapter 2. We'll have a lab 1 today in the lab, but that I'll talk about later at 6:30. Any questions about what we're doing? If not, let's move in. Oops, didn't mean to minimize that. Just trying to get it so I have control here. All right, so we talked about microorganisms and disease and discussed that some microorganisms are pathogenic. That means if they're pathogenic, that they're capable of causing disease. And that's actually a small percentage of microorganisms which are pathogenic. Most microorganisms do not cause disease. They're not pathogenic. And did I talk about infectious versus inherited diseases?
No.
All right. An infectious disease is one that is caused by a microorganism. And can somebody give me an example of an infectious disease? Come on. Say again. We'll go COVID-19. Yes.
Coronavirus.
Coronavirus, that's another infectious disease. Okay, those are all good examples. How about an inherited disease? What's an example of an inherited disease?
Parkinson's disease.
Yeah, Parkinson's disease would be one. Another one that you should have covered in your introductory class would be, sorry, I'm losing it, sickle cell anemia, an inherited disease. An inherited disease is one that you're born with, and it's caused by defective genes. Okay, any question about that? So the sickle cell anemic person had 2 genes, one from their mother and one from their father, that were defective and gave them the sickle cell anemia. Now you can have one defective disease and then you have something called like sickle cell trait. And most of the time those individuals are normal. But if you put them under stress, their cells will sickle. Now, in most of the United States, the people who have one defective disease with sickle cell anemia, most of the time they're normal in most of the United States. But there was an individual who was in Florida, and I think he was a teenager sent to he was a delinquent teenager sent to a camp, and they were making him run first thing in the morning. He didn't have anything to drink, and he was doing all this running in the hot weather of Florida in the summer, and his cells started sickling, and he wasn't able to stay on his feet, and people thought he was faking it, and they took him to the nurse, and she gave him something like smelling ammonia or something like that, and it just caused all of his cells to sickle, and he went into sickle cell anemic disease. I think he died from lack of oxygen. And like I said, normally his cells would have been fine, but he was in Florida in the heat of the summer. He hadn't had any water in the morning, so he had gone all night long without water. And then he was under stress, And then the final straw that broke his back was that they were giving him smelling salts or smelling whatever. And it just was too much for his system. And then all of the cells started sickling. Any questions about infectious and inherited diseases? In this class, we will be mainly talking about infectious diseases because we're talking about microorganisms which can cause disease, and that's just the focus of this class because most of you are going to go into the healthcare profession, and so that is the focus of this class. Any question about any of that? All right, microorganisms are shown in this picture. Let me blow that up a little. And as you can see, they come in various sizes and shapes. They're all small. How small are they? Well, this, if you can see that, we probably can't see that. Let's see if I can blow that. Not easily because it's being hidden. Let's see if I can move that down here. I can only blow this up from a little bit more, but there, you can actually see that there if anyone can read it. I can't read it with my eyes, but can somebody read what that is? Oh, there it is there, 1 micrometer. So that's about 1 micrometer long. That one might be 2 micrometers long and less than 1 micrometer wide. These are 50 micrometers. And that's this one I can actually read, scanning electron microscope. And so you can see that picture there. Different shapes, different sizes. That's actually a white blood cell. And we can see this is 10 micrometers right there from there. So this is, oh, maybe a little, at least 50 micrometers, maybe 10 micrometers long. And then less wide. This here being 50 micrometers from here to here. So that would be a little more than 50 micrometers. And actually, a cell is each of these dots here. And this is actually one colony. That's the mother volvox. And these right here are baby volvoxes that the mother has made. And this will open up and then the babies will go out and float elsewhere. But there's 1, 2, 3, 4, 5, 6 babies inside the mother. And like I said, one of those dots is actually a cell. And the mother is, oh, at least 500 micrometers, maybe 1,000 micrometers long. And then there's another microorganisms. This might be a T cell. I can't see what that's saying, but that's 15 micrometers from here to there. So this is actually the biggest picture. The point is that microorganisms are small. A micron from this size to there is about the size we see under the light microscope with the highest magnification. So that's about the size we can see under a light microscope, and any smaller than that, I think you can get down to, I used to know this, let me see, I think it's 0.2 micrometers is the absolute smallest size you can see in a light microscope. And we'll talk about that later. Any questions on any of this? Let me end this. The magnification. All right, no questions, so let's move on. Microorganisms play beneficial roles, including they decompose organic waste, like when a tree dies in the forest, or when you flush your toilet, that water will go to a waste treatment plant. And actually, it's the microorganisms which are decomposing the human waste. Our microorganisms are producers in the ecosystem. You all know the green plants are producers. Well, microorganisms can also be photosynthetic. Cyanobacteria is an example. This is actually a bacteria which is photosynthetic. It's not green, it's actually blue-green, and they call it blue-green algae. Cyanobacteria is actually bacteria, but it's photosynthetic. And then there are some producers which are chemosynthetic. We'll talk a little bit more about them later in the term. They make their energy needs not from the sun or light, the photosynthetic organisms, but from a chemical. And then they strip electrons from this chemical to make their own food, which would be most likely glucose. Microorganisms I mentioned were involved in the nitrogen cycle, and most of the steps in the nitrogen cycle happen because of a microorganism doing that step. We can use microorganisms to make industrial chemicals like ethanol, which is put in your car, especially I think it's this time of the year, the winter. Most states will put ethanol in the car because it helps remove water from the gasoline. We use microorganisms to make other chemicals like acetone. We use microorganisms to make insecticides, and I'll talk a little bit more about that. This is a different way of getting the insecticide from man-made chemical product, which all of those are pretty toxic. Some of the insecticides made by a microorganism are natural, of course, and they're much less toxic. We use microorganisms to make fermented food, such as vinegar. Let me see, I think all cheeses, if it's a real cheese, bread, if it's got leavening in it, and especially sourdough bread, beer and wine, and many other foods we'll talk about a little bit later. Any question about any of that? We use microorganisms to make products used in manufacturing, such as cellulase. We use microorganisms to make some antibiotics, like penicillin G. And then some antibiotics, we will use the microorganisms to make the first derivative of the antibiotic. And then we will chemically modify that antibiotic to make the final antibiotic. And let's see, ampicillin is an example of that, where they use the microorganism to make the antibiotic, and then we chemically modify it to finalize it as the antibiotic. There are some antibiotics which do not use microorganisms. Can anyone give me an example? I think carboxycillin is totally man-made.
What about clindamycin?
There are others. I don't know about that one in specifics. All of the sulfur drugs are man-made, at least as far as I am aware of all the sulfur drugs. But you're probably right on clindamycin. There are others which are man-made. There are different terms, and I'm just going to mention one of them. We can call it a synthetic or man-made antibiotic. And then there's semi-synthetic. That is one that the microorganisms makes, and then we modify it. And then there's ones that the microorganisms make themselves, and you don't need to know those terms. There are other products that microorganisms use, like today, insulin is made by a microorganism. Initially, when they came up with insulin, they of course obtained it from humans. I'm not sure how they got it from humans. I just don't know the story. They didn't grind up the human, meaning they didn't kill humans, and even from sheep. They then got it from sheep And they didn't kill the sheep to get it either. I'm assuming that they got it directly out of the blood of either the human or the sheep. And then they started growing it in yeast. And I think now they're growing it in bacteria. So now it's totally made by a microorganism. And I know that because my father was given insulin. And initially he was given human insulin, and then he was given sheep insulin. And then he was given insulin made by yeast. And then he was given insulin made by bacteria. Any question about any of that? Well, the making of insulin is a part of biotechnology, which is the use of microbes and organisms to produce foods and products which humans use. Biotechnology is actually centuries old. Most people don't think of it as being centuries old, but something like fermenting grape juice to make wine is actually using biotechnology. The making of sauerkraut using biotechnology. And then the making of insulin, which is not centuries old. That's, oh, when did they start that? Maybe the 1970s or 80s. And that's biotechnologies. What you're thinking of is something being new, we call genetic engineering, a new technique of biotechnology. So genetic engineering is biotechnology, but it's a specific subset. And we use genetic engineering to produce a variety of proteins from bacteria and fungi. For example, insulin made by bacteria, and that's done by genetic engineering. You took the human insulin gene, put it into bacteria, I think it's E. coli, and then got it expressed in E. coli, and the E. coli make the insulin, and then you harvest it from the E. coli. That's genetic engineering. A gene therapy where a child is born with a defective immune system, and you get the correct human gene, which corrects for that defective gene, and you put it into the child. I think we'll discuss that a little bit later in the term. And that's gene therapy. And then there's genetically modified bacteria or genetically modified products, which are using genetic engineering. The flavor savor tomato was made by genetic engineering. I don't know where they got the gene from, but it was a gene that kept the tomato from spoiling rapidly. And so once it became ripe, the tomato would stay ripe for like 2 weeks without spoiling. And that was the flavor savor tomato, which you don't see many of those in the store anymore. So I'm assuming it wasn't very popular. And there's other genetically modified bacteria. As I mentioned, we make insulin, human insulin that way. Any questions about any of that? All right. Microbes are engaged in microbial ecology, where they change the ecosystem. We already mentioned that bacteria and other microbes are involved in the nitrogen cycle. And it's because of the bacteria and the microbes that nitrogen is recycled on the planet Earth. Well, it's more than just the nitrogen cycle. It's also the carbon cycle is running partially because of microorganisms, the sulfur cycle and the phosphorus cycle. All of these elements are being at least partially recycled because of microorganisms. And then the nutrients are also being recycled because of microorganisms. For example, when an organism dies, the nutrients in that organism will be decomposed and recycled by fungi or another microorganism, a decomposer that's either bacteria, archaea, bacteria, or fungi. And then when it's recycled, it can be reused by plants and animals. These are microbial mediated biochemical cycles, the carbon, the nitrogen, the nutrients, the sulfur, and the phosphorus that help support life on Earth. Without microorganisms recycling these elements, life on Earth would be much more difficult because the nitrogen wouldn't recycle. The first understanding of microbial ecology happened by Winogradsky and Bajorink way back in the 1880s to the 1892s. And they were the first to show that microorganisms recycled nitrogen and sulfur in the ecosystem. So they actually discovered that. And we might talk a little bit about Winogradsky columns later. The reason why they were able to show this is because unlike other microbiologists who were studying a pure culture, Winogradsky and Bajorink were actually studying ecosystems of different microbes, meaning many different species come together to recycle nitrogen in the Earth's atmosphere. Any question about that? All right, I'm moving on because I'm not getting any questions. Humans actually do use microorganisms when they engage in bioremediation. Bioremediation means using life to remedy, such as using living organisms to deal with an environmental problem. Like this is an oil spill, and they're spraying microorganisms on them to help break down the oil. And we can do this bioremediation to break down sewage. We've already talked about that. But other pollutants like oil, and we can actually even do it with pollutants like mercury. You can treat the mercury with a live organism, a microbe, and it'll convert the mercury to something less toxic. Now, it's still going to have mercury in it, so it'll be probably some organic form of mercury, but it'll be less toxic than elemental mercury. Any questions about that? Of course, when the oil is broken down by the microbes, it'll break the oil down into carbon dioxide and water. The water will just enter the ecosystem, so that's not toxic. And the CO2 is not terribly toxic, although it could lead to global warming. You would rather have the CO2 release than have the oil around, because the oil is definitely toxic. Any questions about any of that? All right, I mentioned that microbes can be used to make biological insecticides, and this is an alternative to a chemical pesticide, where the insecticide can kill insects and prevent damage to crops or prevent disease transmission. An example is Bacillus thuringiensis, which this bacteria is actually fatal to many insects. And yet this bacteria is harmless to humans and animals and plants. So we can put this bacteria on plants to protect the plants from insects. And this has been available since the 1960s. My father, way back in the 1960s, bought the bacteria to put it on the apples to prevent them from getting worms. The trouble was he put it on the apples and then the rain washed it away. So you need to reapply it on the apples after the rain, and the rain is pretty common in Western Oregon and Western Washington. And you also have to treat the trees every 2 weeks, which is the same thing you did with the insecticide you spray on the tree. You have to treat them every 2 weeks. My father was far too lazy to treat it every 2 weeks and after every rainfall. So the apples did have some worms, but they had fewer worms than if he never put any Bacillus thuringiensis on them. Okay, any questions about any of that? And the humans could eat it. In fact, we did eat the apples, and the Bacillus thuringiensis didn't do any harm to us. And then when my father applied it to the trees, he didn't get any harm from breathing it in. But when you spray the tree with an insecticide, you breathe it in, and that is harmful to yourself. And actually, when I was a teenager, I started spraying the trees. And so I was actually exposed for many years to insecticides. And I have to look that up, what the risk is. Although I was very careful, I always sprayed, let's see, do we say that downwind? No, upwind. So the wind was blowing the other way. So I didn't get very much, but you always get a little bit. So Bacillus thuringiensis is much safer. All right, any questions about microorganisms and their importance in the ecosystem or their importance to humans? No? All right. Let's switch gears and then start talking about the naming, classifying of microorganisms. The classification system, as well as the naming system, was started by this man. Does anybody remember from your preliminary class who established the naming system as well as the classification system? Here's his picture right here. Come on, I can't believe nobody knows.
Carl Linnaeus.
Yep. There's his name actually right there. And he liked Latin names so well, he actually Latinized his name to Carolus Linnaeus. I think his real Scandinavian name, I think he was Swedish, is Carl Linné. And everybody calls him Linnaeus because he liked Latin so much that he Latinized his own name. He actually started his naming system in 1735, and it's still used today as a scientific nomenclature. Every organism has two names. They have a genus name and a specific epithet name, which you need two of them, the genus and the specific epithet, to name each species. Like Homo sapiens discusses humans, that's the human species. Homo is our genus, and sapiens is the specific epithet. Any questions about that? It's called the scientific nomenclature because science uses it. And wherever you are in the world, you can use it to name the creature because it's one name, Homo sapien, which is used in every language for humans. It's also called the binomial nomenclature because every species has two names. Binomial means by two, and nomial means name. Any questions? Sorry, I have something that's a little bit chewy in my mouth. Scientific names are italicized, or if like you're writing it and you can't write italicized, you then underline it. The genus name is always capitalized, and the specific epithet is always in lowercase. So Homo capitalized, sapiens lowercase, and that's supposed to be italicized. It doesn't, yeah, that's italicized or underline. It's always Latinized. One word, Homo sapien, or I should say one name for each species. You use that name everywhere in the world. The name may be descriptive or honor a scientist. Homo sapiens is actually descriptive. Homo means they walk on two legs. Sapiens means wise man. So it means wise man who walks on two legs. Escherichia coli is both descriptive. Coli means it's found in the colon and the intestine. Escherichia is honoring the scientist Esther, how do I ever say that? Who was an early microbiologist. Staphylococcus aureus, another term. This is totally descriptive. Staphylococcus means coccus, a ball. Staphylo meaning ball. Staphylococcus meaning balls in the form of a grape cluster. Aureus meaning golden because they have a golden appearance. They're golden in color. After the first use, the scientific name may be abbreviated with the first letter of the genus and the specific epithet. So normally, Staphylococcus aureus, the first time you use it, you spell it totally out, but all other times you use it, you can abbreviate it S period aureus. Escherichia coli, it's so well known. Most people call it E. coli all the time. But in a scientific paper, you should first call it Escherichia coli, and then later call it E. coli. Okay, but this one is so common, everybody just calls it E. coli. Staphylococcus aureus is found on the human skin and in the nasal passages, and it's not always found on everybody's skin. It's actually only 20% of Americans have it on their skin. It's actually a human pathogen. So if you have it on your skin, you're more likely to get boils. And so if you're a person who gets boils, you might have Staphylococcus aureus, and then you should treat that boil. And if you don't ever get boils, you probably don't have Staphylococcus aureus on your skin. Like the last I checked, I didn't have Staphylococcus aureus on my skin. E. coli is found in people's intestines. It helps us digest our food in both our intestines, the large intestines, or the colon. And then we classify microorganisms to group them in ever larger groups. And that's just to put similar organisms together, like E. coli and Staphylococcus aureus are put in the kingdom bacteria. And then the super kingdom prokaryota. And so we just group things, and that's classifying organisms. Before we can address this, we first need to look at the classification of all living organisms on Earth. And all organisms, when we first break them into categories, we can break them into the 3-domain system. So all living organisms are broken into the 3 domains, and that's the domain bacteria, the domain archaea, or the archaea bacteria, and then the domain eukarya. The domain system is based on the genetic sequence of the organism. So it's based on the genetics of the organism, and the sequence of DNA for all the bacteria is more similar to each other than to any organism in the archaea and any organism in the eukarya. And the 3-domain system is sometimes called the tree of life because it branches like a tree, and it's a tree with 3 trunks. I know that's a very rare tree, but if you think of a tree with 3 trunks, it sort of looks like a tree with 3 trunks. And then it branches off of that trunk. So whenever we discover a new species, we can sequence it. And by sequencing the DNA, we can discover whether it's a bacteria, an archaebacteria, and a eukarya. Any questions about that? All right, I'm not hearing anything. Can somebody confirm that you guys are hearing me?
We can.
Okay, great. Thanks for confirming. Anyways, the 3-domain system is defined. There's no argument about where an organism goes because you can just sequence the DNA and then determine if it belongs in bacteria, the archaea, or the eukarya. In the other lower classification systems, we don't have it defined. Like with the kingdoms, which comes underneath the domains, there is no definition for what is a kingdom. We can say that the bacteria have only one kingdom, so the kingdom is the same as the domain, bacteria. And the same with the archaea. The kingdom is the same as the domain. But the eukarya can be broken down into plants, the plantae, the animalia, the animals, the fungi, and then the protista. And there is no definition for what makes a plant, what makes an animal, what makes a protista. And in fact, some people actually argue, do the large algae, are they protista or are they plants? And because there's no good definition, that's a point that some people argue. Euglena has some properties of animals. It moves around and can eat things. And it has some properties of a plant. It can be photosynthetic under light and conditions where glucose is not around. It'll be photosynthetic. But then it also has properties of a protista. Okay. So what is it? Well, there's arguments about what it is, and that's just the case. We don't have a good definition, especially for what a protista is. A protista is defined as a eukaryote that is not a plant, not an animal, and not a fungi. So that's all other eukaryotes. And as you can imagine, the protista is not a monophyletic group. And there are arguments for how many kingdoms there are. We're going to use the 6-kingdom approach. 1, 2, 3, 4, 5, 6 kingdoms, meaning counting bacteria and archaea. But other people use the 5-kingdom approach. And let's see, that's joining archaea and bacteria into one kingdom, and then plants, animals, fungi, and protista. Other people use the 3-kingdom approach, and that's the same as the domains. Some people use the 10-kingdom approach, and there's bacteria, archaea, plants, animals, fungi, and then they break the protista down into different kingdoms. And you can break this into the 10 different kingdoms, the 15 different kingdoms, or even the 20 different kingdoms. Like right here, let me try and show you. Pull this up a little. We could break the protista into this one, that 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 one. Not these, because those will be plants, fungi, and animals. But you could break the protista into 1, 2, 3, 4, 5, 6, 7, 8, 9 different groups. Okay. Any question about that? And then other people have higher kingdoms. They start breaking down the bacteria into different groups and the archaea into different groups. And so there's the 20 different kingdom approach, the 30 different kingdom approach. And there's actually an argument among biologists for the number of kingdoms. We're going to use just simply the 6-kingdom approach because it's easy. And I think it's what your textbook uses. And hopefully that's what your textbook used in your preliminary class, Biology 160, if you took it at Clark. But there are arguments for how many kingdoms there are, and the scientists themselves don't agree. And they've been arguing about that for 20 years. So I don't think it'll ever be resolved. Any question about that? You can then break down the kingdom into different categories. We're not going to break it down. Let me see what comes after kingdom. King, Peter's, son, king. So class, order, phyla. We're not going to look into that. There is one category that you should know, and that is what your book calls a cell type. The bacteria have prokaryotic cells, and prokaryotes is a cell type. Archaea also have prokaryotic cells, but eukarya have eukaryotic cells. So the cell type is eukaryotic. Now you could call these super kingdoms and to do the division that way, meaning you have 2 super kingdoms. Your textbook doesn't discuss that, so I'm not going to say anything further about it. We'll just call it a cell type because that's what your textbook calls it. And you just need to know the archaea and the bacteria, although they're 2 different domains, they have the same cell type. And the eukaryotes have 1 cell type, the eukaryotic cells. Any question about that? You should know that when people first started sequencing microorganisms, that they thought the archaea and the bacteria belong to the same kingdom, the same domain. Okay? Because the archaea were prokaryotes and so were the bacteria. But when we sequenced them, we found the archaea who had different DNA than the bacteria. And that came as a surprise to biologists in the 1970s. Any question about that?
You said that we need to know which cell type belongs with which domain. Is that what you said?
Yes.
Okay, thank you.
Okay. All right, let's talk about the domain bacteria. It'll be the main focus of this class, actually. And we'll talk a little bit about why that's the case later. They are prokaryotes. They do have peptidoglycan in their cell wall. So that's something we can use to distinguish them because no other cells have this chemical, peptidoglycan. And we'll talk a little bit more about what that is. Most of the cells in bacteria divide or multiply by binary fission. That's simply the cell divides into two. For energy use, the bacteria can get their energy needs from all of the sources. Some bacteria get it from an organic chemical, and that's what we do. We get it from the organic chemical in the food we eat, largely glucose. And that's what most bacteria do, like E. coli will break down glucose or some other organic chemical to get its food or energy needs. But some bacteria can get their energy needs from an inorganic chemical, such as hydrogen sulfide or a sulfate or iron. And then other bacteria can get their energy needs from photosynthesis using light and carbon dioxide and water to make their own food. So in reality, they're getting their energy from light. Most of the domain bacteria are free living decomposers or primary producers. So most of the bacteria either degrade something or they are a primary producer, making their own food. And that would be like the cyanobacteria or the chemosynthetic bacteria. Some of the bacteria are parasitic and some of the bacteria are pathogenic. Pathogenic, once again, remind you, is a bacteria that causes human disease. An example of a pathogenic human bacteria would be Staphylococcus aureus. All right, the domain archaea, let's talk a little bit about them. All of their cells are prokaryotes. Does anybody know, do the archaea have peptidoglycan in them?
No.
Yeah, no, I just told you a little while ago if you didn't know. They do not have peptidoglycan. Most of the archaea do not have peptidoglycan. And we're not going to talk about the exceptions, so we'll just say no, they do not. Some of them are found in extreme environments. And when we first found the archaea, it was bacteria growing, or I shouldn't say bacteria, it was prokaryotic cells growing in extreme environments. But when we look, well, let me talk a little bit about the extreme environments. It could be the methanogens living in environments where there was methane. They produce methane, the methanogens. It could be the extreme halophiles, a bacteria living in a high salt concentration, like the Dead Sea or the Great Salt Lake, an extreme halophile. It can be an extreme thermophile, an organism living at very high temperatures in a, what do you call it, a hydrothermal vent. But when we look further, we can find the archaea in all of the Earth's biomes. And they're a significant percentage of the life in that biome. So you can find them a certain percentage of every Earth's biome. They're especially common in the soil and in ocean water. There'll be plankton, a significant portion of plankton. Does anybody know, or any of the archaea pathogenic?
No.
Yes, you're correct. Good job, by the way, because I haven't talked about this. The archaea, there's no species of archaea known that causes human disease. Okay, so they are not pathogenic. And we'll briefly talk about the domain eukarya. Let me remind you, we can split them into 4 different kingdoms. The kingdom Plantae, the kingdom Animaliae, the fungi, and the protista. All of these cells lack peptidoglycan. So if a cell has peptidoglycan in it, it is going to be from the domain bacteria. Any question about that? The kingdom fungi actually have chitin in their cell walls. You don't really need to know that. They use an organic chemical for getting their energy needs. And fungi always decompose something. You can have multicellular molds in the fungi. And this is showing you a multicellular mold, something like bread mold. You can have mushrooms, which are also multicellular. And they are composed of masses of mycelia, which are composed of filaments of hyphae. So here we're seeing a hyphae. But some of the molds and all of the yeasts are unicellular. And they are either decomposers or pathogens. So the yeast either decompose something or cause disease in an animal or a plant. And here we're looking at a yeast. Bread yeast. Is it a decomposer, a pathogen, or something else? The yeast you use to make bread. Is it a decomposer, a pathogen, or something else?
Is it a pathogen?
No, it doesn't cause human disease.
Something else?
No, it's a decomposer. It's a good guess that it's something else because we're using it to make bread, but it's actually decomposing the flour and the sugar in the bread. So that's why it's called the decomposer. Okay.
What about nutritional yeast?
Oh, that's yeast that you're eating for nutrition. Well, while it is growing, it is a decomposer. Okay. But when you're buying it, it's no longer growing. And if it's growing, it's acting as a decomposer. Okay. But most people who get nutritional yeast, they don't grow it. You could grow your own yeast and you can use it to make sourdough bread. Of course, there will be bacteria in the yeast culture, in the sourdough culture. So now sourdough culture is actually both yeast and bacteria to make the sourdough. You don't need to know that, I'm just telling you. All right, the kingdom Protista, let me remind you, is a eukaryote that doesn't fall in the plants, the animals, or the fungi. In the Protista, some of them are plant-like protists, and these are the algae. They have cellulose in their cell walls. They engage in photosynthesis for their energy needs. For a lot of them are green, but the algae that blows up on the Oregon and Washington coast, the brown kelp, it is an algae, but obviously, and it is photosynthetic, but it isn't green because its photosynthetic pigment is not chlorophyll. Its photosynthetic pigment is something that's brown or red in color. And the algae produce oxygen from photosynthesis. And it also makes organic compounds from photosynthesis. And that would be, you take carbon dioxide and water and first make sugar, and then it can make other organic compounds from sugar. But sugar is an organic compound, so we can stop right there. Any question about the algae? The kingdom Protista also includes the protozoa, and these are the animal-like Protista. Those include the amoebas, the paramecium, euglena, and here we're looking at an amoeboid cell. It's not a protista, but it's an amoeboid cell. It moves by pseudopods, and wherever the pseudopods are being put down and added to, the cell contracts elsewhere and then moves that way. The protozoa either absorb or ingest organic chemicals. They may be motile with either pseudopods, cilia, or flagella. Does anybody remember if a bacteria is motile, what does it have to be motile? Do bacteria have pseudopods?
No.
No, that's correct. They don't have pseudopods. They don't have cilia. So if a bacteria is motile, it has flagella, or at least one flagellum. But eukaryotes can be motile by all three of these. And we're talking about a microorganism can eukaryotic cell can be motile by all three of these. Of course, I'm motile by my legs moving, and that's not because of pseudopods, cilia, and flagella. It's because of the, what's that, flagellum? That's not right. Myosin, yeah, it's myosin, the protein that contracts my muscles. And we're not going to talk about that. And there's a third group of the protista, and that would be the fungus-like protists, and we're not going to talk about them. An example would be a water mold, which actually caused the Great Famine in Ireland. When was that? In the late 1800s or the early 1900s? And we're just not going to talk about them. The plants include mosses, ferns, conifers, and flowering plants. So we can divide plants into mosses, ferns, conifers, and flowering plants. All plants are multicellular. All animals are multicellular as well. So if it's animal-like or plant-like and a eukaryote, it will be a protista. Because if it's unicellular, it can't be a plant, it can't be an animal. The animals include sponges, worms, we can call that a helminth. And we'll talk a little bit about them later. It includes insects, other crustaceans, and then the vertebrates. Any question about that? All right, let's talk about what a microorganism is and what comes underneath the umbrella of a microorganism. All the bacteria are microorganisms. All the archaea are microorganisms. All viruses and prions are microbes. They're not organisms because viruses and prions are special. They do not have cells, but they have some properties, not all properties of a living life form. And so you could argue they're alive, but they don't have all the properties of a living life form. The main one that's missing is they do not have cells. Certain of the eukarya are microorganisms. The protozoa, all of them are eukaryotes. The microorganism. So all of the protozoa are microorganisms. All molds, all yeast are microorganisms. Many molds, but not all molds, are microorganisms. Some algae, if the algae is unicellular or very small, it is a microorganism. And then the helminths, the worms, are also a microbe, a microorganisms. And how come that is? Worms are not microscopic, right? So why are we including the helminths as a microorganism?
Is it because in some of their life stages they're microscopic?
Yes. Let me turn up the heat here. It's a little cold. For some reason. Yeah, it probably just needs to turn on. It hasn't turned on. Some of the life stages of the worms are microscopic. In fact, the stage of the worm that is infective is usually microscopic. So microbiologists study organisms that cause human disease, and some worms cause human disease. And the worms that cause human disease, the stage of the worm that causes disease, the fertilized egg usually, is microscopic. And so we do include the helminths, the worms, as microorganisms, even though the adult worm usually is not microscopic. There are a few species where the adult is also microscopic, but all the other species, the adult is not microscopic. For the, let me see if I can remember the name. For the human, oh gosh, I just had it. Not the trematode, not the nematode. The human flatworm that causes intestinal distress. I'm trying to think of what the name of that is now. I'm drawing a blank there. Tania saginatum, which is a flatworm. It's a cesto. That's what I was trying to think of. The scientific term for a flatworm is a cesto. Those are parasitic. And the one that I mentioned actually gets up to, let me think now, is it 30 meters long? Something like that. And so it's obviously very, very large. And what it is, it's typically long and skinny, and it can be all of your lower intestines. It can be the length of your entire lower intestines. And that's why it's so long. You should realize that the helminths, although many of them are parasitic, they're not all parasitic. The adult stage is not even microscopic. It includes the flatworms and the roundworms. And these worms, at least the parasitic worms, include the flatworms and the roundworms. And these two worms are very different from each other. They're in different phyla. And they're as different from each other, a flatworm from a roundworm. A nematode would be a roundworm. I'm trying to think. tapeworm, like Tania saginata, is a flatworm. And then, what is it? The disease you get in pork. Trigonosis is a roundworm. And they're as different from each other as they are from us. So the flatworm and the roundworm look similar to us, and we call them a worm, but they are genetically as different from each other as humans are from the flatworm. Okay? Any question about any of that? All right, no questions. Let's move on. Briefly talk about viruses. They are special in that they are a microbe, but they're special because they have no cells. They do have either DNA or RNA as their core molecule inside them. And that DNA or their RNA is their genome. But viruses never have both DNA and RNA in the same virus. Any questions about that? The virus will have DNA and RNA, and then surrounded by that, you'll have a protein coat around its nucleic acid. That's made of protein, by the way, the protein coat. The coat may be enclosed within a lipid envelope, but not all viruses have a lipid envelope. Only some of them do. And viruses only replicate when they've infected their host cell. So when a virus is outside of its host cell, the virus cannot replicate. Viruses are not part of the 3-domain system. So when we divide all organisms into a domain, we're not talking about viruses there. Any question about any of that? If there's no questions, I'm going to stop here. And I will start here on our next lesson. And I'll see you at 6:30 for the lab.